Dear colleagues, I want to present the research title as Testing the States of Erythrocytes under Hypertonic and Posthypertonic Shock. Cryopreservation provides long-term storage of biological specimens with subsequent restoration of their functions after thawing. Storage of erythrocytes at ultra-low temperatures does not lead to their damage, because under these conditions, cellular metabolism is inhibited. Cell destruction occurs during the stages of freezing and thawing due to the action of cryodamage factors. To determine the contribution of each of them to the total cryodamage of cell, model experiments are used. Hypertonic shock of erythrocytes simulates the effect on cells of cryodamage factors acting at the freezing stage, whereas the effect on erythrocytes acting at the thawing stage, studying using post-hypertonic shock. In the base of cell damage under action of temperature osmotic shock, there are processes associated with transmembrane pore formation. Therefore, the use of membrane-tropic substances is advisable. Research aim was to study the development of hypertonic and post-hypertonic hemolysis of human erythrocytes at 37 and 0 Celsius degree in the presence of chlorpromazine and trifluoroperazine. Hypertonic shock was performed by transferring erythrocytes into 4 mole per liter sodium chloride at 37 or 0 Celsius degree. Post-hypertonic shock was initiated by transferring erythrocytes from dehydration medium into rehydration 1 and 37 and 0 Celsius degree. The cationic amphiphilic compounds were used, chlorpromazine and trifluoroperazine. The amphiphilic compounds were added into media prior to transfer the cells. The level of erythrocyte hemolysis was determined by spectrophotometric. The maximum antihemolytic activity was calculated by this formula. The morphological features of the cells were elevated by light microscopy. From this slide, it can be seen that hypertonic hemolysis level of human erythrocytes depends from temperature, and it's 90% at 37 Celsius degree and 60% at 0 Celsius degree. While the level of post-hypertonic hemolysis of erythrocytes depends on temperature to a lesser extent. However, the level of erythrocyte hemolysis at 0 Celsius degree was lower. It can be seen that the usage of chlorpromazine and trifluoroperazine leads to a decrease in the level of hypertonic hemolysis erythrocytes at 0 and 37 Celsius degree. The presented dependencies are characterized of 3 areas. First, a decreased erythrocyte hemolysis level is observed. Then a plateau of effective concentration is formed, and a further increased concentration of amphiphils leads to a cell damage. It can be noted that the plateau of concentration at 37 Celsius degree are shifted towards higher values than at 0 Celsius degree, in a case of usage both amphiphilic compounds. From this slide, it can be seen that amphiphilic compounds doesn't reduce the post-hypertonic hemolysis level of erythrocytes at 37 Celsius degree. However, at 0 Celsius degree, these compounds decrease the erythrocytes hemolysis level. It should be noted in case of chlorpromazine, the concentration plateau are shifted towards the higher concentration than it's for trifluoroperazine. From these dependencies were determined values of effective concentrations and calculate values of antihemolytic activity, which are presented in this table. Under hypertonic shock, antihemolytic activity of both amphiphilic compounds is about 2 times higher at 37 Celsius degree. The efficiency of amphiphils at post-hypertonic shock erythrocytes observed only at 0 Celsius degree. Comparative analysis of efficiency of cationic amphiphilic compounds show high anti-hemolytic activity of chlorpromazine compared to trifluoroperazine. And conversely, under hypertonic shock, values of anti-hemolytic activity for trifluoroperazine higher exceeded similar indexes for chlorpromazine. But for both types of shock, the effective concentration of chlorpromazine are higher than the ones of trifluoroperazine. It is known that cationic amphiphils integrating predominantly into the inner monolayer of erythrocytes membrane cause stomatocytosis. Signs of stomatocytosis are observed even after the cells were exposed to posthypertonic shock using amphiphils at effective concentrations. Amphiphilic compounds, due to their amphiphilic nature, incorporated into the membrane and prevent hemolytic pore formation. Since the protective effect of amphiphils is realized under post-hypertonic shock only at 0 Celsius degree, and under hypertonic shock compounds are more effective at 37 Celsius degree, the mechanism of action of amphiphils under hypertonic shock can be assumed to be associated with perturbation of membrane, while under post-hypertonic shock it can be due to this stabilization. Thus, research shows different efficiency of cationic amphiphilic compounds under two types of shocks, which simulate the effect on cell of damage at the freezing and thawing stages. This may indicate the different nature of the mechanisms of formation and development of hemolytic pores under these conditions. I would like to thank Igor Chabanenko for performing microscopic examination, and thank you for attention.
Thank you, Elena, for such a nice talk. And we have now time for a couple of questions. It's really exciting to see that hyper and hypertonic shock depends on temperature, and the cells do not respond similarly under the different temperature. How, by the way, you can explain it, that you have absolutely different results when you vary the temperature, and the osmotic shock of cells is strikingly different?
Under hypertonic shock, pore formation in the membrane depends on temperature and is prevented by incorporation and distribution of amphiphils in the membrane at 0 and 37 Celsius degree, and effect more expressed at 37 Celsius degree. The perturbation of erythrocyte membrane both as a result of incorporation and increase the temperature and successfully prevent the formation of hemolytic pore under condition of hypertonic shock. And conversely, under post-hypertonic shock, pore formation is slightly depends on temperature and prevent by incorporation and distribution of amphiphils in the membrane only at 0 Celsius degree. So in this case, the effect of amphiphil incorporation and low temperature were multidirectional. Thus, some stabilization of erythrocytes as a result of lowering the temperature to 0 Celsius degree allow amphiphil to prevent the formation of hemolytic pore under condition of post-hypertonic shock.